DNA has all the secrets of life and contains within it the purpose of life, which is to survive and reproduce. DNA contains the coding for proteins, which are how we function, including enzymes, so they regulate metabolic processes, and it's also how you pass information from one generation to another. So the discoveries involving DNA were very monumental in understanding biology, including many of the fundamental theories such as cell theory and the theory of evolution. So some of the first basic experiments done with or about inheritance include those done with microbiology. So Frederick Griffith in 1928 looked at this strain of cells which were infected with a disease which caused mice to die. However, he was able to treat these cells to remove their outer covering, which protected them. And when he did that, he created this separate strain called R cells. And when you gave those to a healthy mouse, the mouse would remain healthy and would not die. You could also heat kill the S cells, and they, when injected into mice, would also leave a healthy mouse. But if you combine the two, a heat-killed and a living R cell that was not lethal before, they would kill the mouse, and you could extract from that mouse living S cells. So there must have been some transfer of genetic information from one, they didn't call it genes at this point, but from one to the other. And this then led scientists wanting to know, well, what was it? So another experiment done by Avery McCoy and McCarthy in 1944 repeated the experiment with Griffith, but used purified cell extracts, including pneumonia. And what they did is they injected the mouse with destroyed DNA and destroyed proteins to see what happened with the experiments. When they destroyed the proteins, the experiments turned out the same. But when they destroyed the DNA, the mice lived. So the conclusion was, well, the genetic information that is being passed must be in the DNA. Okay, so DNA was the front runner at this point for what had the code, or what was being passed from one to the other. Another experiment then in 1952 with Hershey and Chase, they looked at bacteriophages. Bacteriophages are viruses that infect bacteria. And they radio-labeled the protein and the DNA and then allowed them to infect the bacteria, and then they let them go for generations and pulled out the parents. And only the radioactive DNA showed up in the bacteria. So that must have been what was being passed. When they did with the protein, that didn't show up in the bacteria. So there's no radioactivity being passed. So the conclusion was, again, DNA is what's being passed from one generation, or one bacteriophage in this instance, to another. The structure of DNA, however, was still a mystery. So we'll talk later about how these things were discovered. But DNA, as we have studied before, is a nucleic acid, which means it has a 5-carbon sugar, a phosphate group, and a nitrogenous base. DNA has a free hydroxyl group, and one of the carbons of the sugar, which identifies it as deoxyribose nucleic acid as opposed to ribonucleic acid. And there are 4 nitrogenous bases: adenine, thymine, cytosine, and guanine for DNA. The phosphodiester bond is what forms between 2 nucleotides to form then DNA, and I have those highlighted there. The ester bonds on either side of a phosphorus thus describe this phosphodiester bond. And this is formed between the 3', so these carbons on the ribose are identified by where they are located. So this is 1', 2', 3'. The carbon from the 3' then binds with the phosphate, and the phosphate, the second ester bond, then binds to the 5' carbon of the next nucleotide. So as a 5' to 3' orientation, that means we're talking about the 5' is on top and the 3' is on bottom, and it goes in that order. Alright, so the next experiment was done by Erwin Chargaff. It wasn't the next one, but one of the most significant ones. And he found the amount of adenine equals the amount of thymine in DNA, and the amount of cytosine equaled the amount of guanine in DNA. And they knew the structural form or the structure of these bases and named them according to their structure. So purines had this double hexagonal and pentagonal structure combined, and then pyrimidines had just the hexagonal structure. So the pyrimines and the pyrimidines lined up together, and so from that came Chargaff's rules, which is adenines bind to thymines and guanines to cytosines. The shape was discovered by a scientist named Rosalind Franklin. She performed X-ray diffraction. Here is one of her pictures to identify the 3D structure as a double helix. And she also was able to discover that it had a diameter of 2 nanometers. It makes a complete turn every 3.4 nanometers. So she didn't understand the inside of this, but the outer layers were understood. All right, this is a helical structure, and this is how frequently it turns. Watson and Crick then, in 1953, deduced the chemical structure based on Chargaff's rules, Rosalind Franklin's work. They didn't actually do an experiment themselves, but they were able to deduce then, based on that information, the molecular structure, made a model of the DNA structure. And from there, it was more of an epiphany that, oh, this must be how information is passed. So you have 2 strands of polymers of nucleotides going in opposite direction with a phosphodiester backbone, and they wrap around one axis, and the bases pair with each other. And so these are this is a figure of the partial chemical structure then of DNA. You have one end going from 5' to 3', and the other one is anti-parallel, meaning it's upside down compared to the other one. So its top would be 3 and bottom would be 5'. The 2 anti-parallel strands then are bound together by the hydrogen bonds which form between the base pairs. 2 hydrogen bonds will form between adenine and thymine, and 3 between guanine and cytosine, and this gives it a consistent diameter. Then looking at replication of DNA, there were 3 possible ways in which it was thought DNA could have been replicated. You could make a copy, and then that copy would be the new strand, and the old strand would keep its same strands. And that would be the conservative model, or it could be the dispersive model, where you have 2 parts that are inherited together, where you have new strand, old strand, new strand, old strand. But the model that actually has the most had the most evidence and seems to be the most and is what works is a semi-conservative model, where one strand is used as a template to make a new strand, and the other strand is a template to make another strand. So this is a semi-conservative model. So DNA replication requires three things. Something to copy, and this is the parental DNA molecule. Something to do the copying, which is done by enzymes, and the building blocks to make the copy, which are nucleotides with ATP on them, or triphosphates. Not ATP, but the energy from the phosphates. And it's broken down into 3 processes: initiation, elongation, and termination. Bases are added to the 3' end of an existing strand, so on this side of this strand and on the other side of the other strand. And they require a DNA polymerase, which is the enzyme which adds the nucleotide to the strand being replicated. DNA polymerase matches existing DNA bases with complementary nucleotides, so again A to T and C to G, and the new bases then add to the 3' end. So if it's 5' over here, and they're reading in the 3' to 5' direction. In order for DNA polymerase to get started, though, it needs an RNA primer. So another enzyme is used to set down the primer. Replication is very similar between eukaryotes and prokaryotes, but E. coli is the bacteria found in your gut, and many other organisms, and it was used as the model species to discover this. As it's a prokaryote, so it has one circular DNA molecule. Replication begins at the origin of replication, and we have studied this before in binary fission, and then it proceeds in both directions. And here is you can see a bubble here formed because it is forming then a second strand. E. coli has 3 DNA polymerases. DNA polymerase 1 acts on the lagging strand. We'll talk about that later. To remove primers, so the RNA primer has to eventually be taken off, and new primer or new DNA should be put on there. DNA polymerase 2 is involved in DNA repair processes. DNA polymerase 3 is the main replication enzyme. And so when we say DNA polymerase, this is usually the one we are referring to. All of them have a 3' to 5' exonuclease activity, so it can read the DNA and help take out mistakes that have been made. And DNA polymerase 1 can also do that in the opposite direction, from 5' to 3'. Helicase then is another enzyme which is important for opening and unwinding the DNA, since it is twisted. And then you have single-stranded binding proteins right here and here, which help the hydrogen bonds, or prevent the hydrogen bonds from reforming and keep them apart. Topoisomerase then is another enzyme which prevents supercoiling. So as you're unwinding it, when you try and unwind things, then that will cause, as you unwind in one direction, it has to wind in the opposite direction. And so topoisomerase keeps that from happening. So because you have 2 antiparallel strands, you can't have continuous replication in both directions. So DNA polymerase, because it can only go in one direction, makes for a semi-discontinuous process. The leading strand, which would be going in the direction it can synthesize, can just constantly add more nucleotides as the DNA unwinds. But the other strand, because it's in the opposite direction, that has to kind of leapfrog itself and makes these fragments called Okazaki fragments. So at the replication fork is where this DNA is opened and replication is allowed to start. DNA primase can then add nucleotides after RNA polymerase makes a primer. And so a DNA primer or an RNA primer will be needed for DNA polymerase only once on the leading strand. As it opens, it can just continually add more. But for the lagging strands, you're going to have lots of them. And then they will be replaced later. At the end, after DNA polymerase is able to remove the RNA primers, there's still a little bit right there, so between these two, that needs to be sealed or bound. So a phosphodiester bond needs to be formed there, and ligase is the enzyme that does that. Gyrase then is the DNA gyrase is one that unlinks the copies once they are completely finished. So this is kind of the finishing touch. Okay, so again, here's that little nick between the two fragments, and DNA ligase comes in and seals that nick and forms that bond. All right, so then here then is the replication fork with all the enzymes. We have a lagging strand which is being synthesized in a leapfrog manner, and the leading strand, which is just adding on nucleotides as this opens up. And here is another example of the leading lagging strand and the different enzymes. We'll go over this in class. All right, so that's replication in a nutshell. The next thing we're going to talk about is how we get a gene to make a functional product, which then carries out the functions of the cell. This is done through what a theory called the central dogma. And the dogma is something that is standard, right? So this has to be done in all living things in order to make proteins. So really what you're doing is you're taking DNA, which is the instructions, you're making RNA, which is a more condensed, specific instruction. DNA has all the instructions, and you don't want to copy everything all at once, you just want a little piece. So you make a little copy of that, say, for a hemoglobin, and then from that little copy you can make a protein. The process of making DNA into RNA is called transcription. As you remember, DNA is a double helix of nucleotides, and RNA is a single strand of nucleotides. And then the second part is translation, where you're taking RNA and converting it into protein. So the way that I remember this, C, they're alphabetical, so the C comes before the L. Transcription is something that you're doing in the same language. So DNA and RNA are both in nucleotides, so they're very similar. Translation is where you're going from one language to another. So for RNAs and nucleotide, well, proteins are made in amino acids. So here you have two completely different things. So you need to translate that information into something different. DNA has adenine, guanine, cytosine, and thymine as its bases, and those are the building blocks. A pairs with T and G with C. RNA has bases as well, but it has uracil instead of thymine. And A pairs with U instead of T in this case. And you have a single-stranded molecule. Protein then, the fundamentals of proteins are amino acids, and there are 20 different kinds, and they have different molecular properties. And it's read from codons of mRNA sequences, which are these triplet nucleotide bases that are read, and then amino acids are added onto them. The final structure then is a polypeptide of a protein, which is a bunch of amino acids put together. So, looking at them all together, all three of them, or both processes, require a blueprint, machinery, and building blocks in order for them to occur. And then the process itself is a series of steps. The blueprint in transcription is DNA. The enzyme is RNA polymerase. Okay, this is what's going to make RNA. And the building blocks are nucleotides, adenine, guanine, cytosine, and uracil. Once those are put together through by RNA polymerase, it starts through the process of initiation, and then continues through the process of elongation, and finally ends with the process of termination. Translation is the same in that it starts with a nucleic acid, but the end is protein. So our blueprint is our messenger RNA that we got at the end of transcription. The machinery or enzymes are done by the ribosome, ribosomal RNA, and the amino acids are the building blocks, which are transported to the ribosome through transfer RNA. So the process, however, is the same steps: initiation, elongation, and termination. So we're going to go through these individually. Here's the figure from your book, again showing our double-stranded DNA as our template. Transcription converts it into single-stranded mRNA, and translation into protein. All right, there are different types of RNA, and each has a purpose in the different processes. Messenger RNA is that blueprint for translation and the product of transcription. Ribosomal RNA forms the ribosome, and transfer RNA is very important in carrying amino acids for the translation process. So Francis Crick and Sidney Brenner determined how the order of nucleotides in DNA encoded for the amino acids, and they learned that a sequence of three nucleotides with their bases then coded for different types of amino acids. And they did this by inserting or deleting a nucleotide to change the base pair sequence. And when they did that, they could determine, it changed that into something else. After doing that enough, they figured it out. So the genetic code was identified by Marshall Nirenberg for each amino acid. There are three codons for stop, UUA, UGA, and UAG. There is one codon that is the start codon, that's AUG, which also codes for methionine. And then there are 20 other amino acids which can be added continually onto each other. The code is degenerate, meaning some amino acids are specified by more than one codon, and this code is practically universal. So the same code is used for all living things. And this then is the table that shows then the bases and the combination and what you get from each. So a CUU forms a leucine. AUG forms methionine and or the start. And then here are stop codons, UAA, UAG, and UGA. All right, so for transcription to start, RNA polymerase adds the RNA nucleotides and combines them together. Initiation occurs when a transcription unit is identified by mRNA. So you have a promoter, which is a sequence which promotes initiation, the start site, and the termination site. And these are all three different sites away from each other. So the promoter site is going to be upstream from the start site and is just where it recognizes, like, okay, then up here somewhere I'm going to have a start site. Okay? And it is not transcribed itself. It just helps. Now, this is not this is an asymmetrical process. So by binding to the promoter, it binds in a specific direction, and so then it knows which way to go after that. Elongation, then, the amino acids are added to the 3' end. So it grows from the 5' to the 3' direction. Sorry, not amino acids, but the nucleotides are added to the 3' end. And in order to do that, a bubble is made where the DNA is slightly unwound. So in this, it's okay if the DNA just closes behind it. So it slightly unwinds, adds the nucleotides, and the messenger RNA is just kind of formed out of the back. Okay, and then after it passes, that closes over itself. So it doesn't need all the enzymes that are involved in replication. Termination is marked by a sequence that specifically says for it to stop, for the polymerase to stop. And this causes the formation of the phosphodiester bonds to cease. The RNA-DNA hybrid with transcription bubble dissociates, and RNA polymerase releases the DNA. In prokaryotes, this is done because of a hairpin, which forms by the messenger RNA. So where the messenger RNA folds over on itself and makes this little mRNA. After messenger RNA is made, it then needs to have some modifications to it. So a 5' cap is put on the 5' end, and a poly A tail, where you have lots of adenines, is put on the 3' end. And then the non-coding sequences are pulled out through a structure called the spliceosome. So you start with a pre-mRNA, so it's actually a lot longer than what you end up with. So what you're going to do is you're going to take introns and pull them out. So this is just non-coding sequences that are not necessary. And I don't know exactly why it does this, but if they are not coded, they are spliced out. So here's a spliceosome here. The exons then are the ones that are going to be spliced together. You have small nucleoprotein particles recognize these intron and exon boundaries, and then these are also called SNRPs, then cluster with other proteins to form that spliceosome, which removes the introns. You can also have alternative splicing. So this allows for one gene to actually code for multiple products because you can move around the exons. So here you have 1, 2, 3, 4 exons, and you can create a protein by putting 1, 2, and 3 in order, and then 1, 2, and 4, or 3, 2, and 1, whatever order is necessary to make the proteins. So this is how 25,000 genes are able to make 80,000 different mRNAs. All right, the tRNA molecules then carry the amino acids to the ribosome to incorporate them into a polypeptide. So this is important for translation. So they first need to be charged. They need an amino acid. So aminoacyl tRNA synthetase adds the amino acids to the tRNA, to the acceptor stem. So this one doesn't have an amino acid on it, but if it goes to the enzyme, it'll get one added to it. The anticodon loop then reads the messenger RNA and corresponds with the amino acid that it has. So each aminoacyl tRNA synthetase recognizes only one amino acid, but there are several tRNAs. This charged tRNA has an amino acid added using the energy from ATP, and then it can undergo a peptide bond formation without additional energy. So it has an ATP on it, and then it can go to the ribosome. Ribosomes don't verify the amino acid attached to the tRNA, so they just assume as long as it matches up with the codon, everything's all good. The ribosome has multiple tRNA binding sites, the A site, the P site, and the E site. The A site is where the new amino acid comes and binds. The P site has the amino acid chain and then passes it on to the A site. And the E site is after the P site has given its chain to the A site, it moves to the E site and then exits the ribosome. So the ribosome has two functional units, a large subunit and a small subunit. The functions then of those two are to decode the mRNA and form the peptide bonds. So peptidyl transferase then is the part of the ribosome that is the enzyme that allows this reaction to occur or increases its ability to do so. And it forms the peptide bond between the two amino acids. So the initiation complex includes our initiator tRNA with our informal methionine or methionine in eukaryotes, the small ribosomal subunit, and the mRNA strand. So this is what first comes together. Then you have the ribosome binding sequence of mRNA, which positions the small subunit in the right spot, and the large subunit is then added on top. Now it is ready to add more to the A site. But the first site, which is filled, is actually the P site. Elongation then adds amino acids. So the second charged tRNA goes to the A site, which is empty. The peptide bond then forms between the two amino acids, and then everything shifts over, and the next amino acid is added. So this occurs through a cyclical event where you have the amino acid chain on the P site, a new one comes in on the A site, the bond forms between the A and the P site, and the amino acid chain goes to the A site. Everything then moves over, the one on the P site exits, and now we are ready for another one to come into the A site. So that goes over and over and over and again until termination, which is where you come to a stop codon, UAG, UAA, or UGA. A release factor then comes in there, and it causes dissociation of the ribosome. So ribosome splits, the mRNA goes one way, the tRNA goes the other, and now you have the polypeptide. There are fewer tRNAs than codons, and that's because this third part of the codon and the anticodon doesn't need to be exact. So that allows for some wiggle room. Wobble pairing allows them, the pairing between the 3' base and the 5' base of the anticodon to not have to be exact. So fewer tRNAs can accommodate all the codons and all the amino acids. Okay, and that's why there is some redundancy in these sequences and what they code for. Alright, so that's replication, transcription, and translation, the central dogma, and now we're going to talk about some things that can go wrong. So mutation is when you have an altered gene, or something in the DNA is different than what's supposed to be. A point mutation alters a single base. Okay, so if this is the normal DNA, RNA, and amino acid sequence, a point mutation is just where one of them changes. A base substitution may be where one is for the other. Okay, so if you get an A here instead of a G, well then that can affect the amino acid, but if it still codes for the same amino acid, then it's called a silent mutation. So nothing was affected there. A missense mutation, which would be this one, is where the amino acid changes. Okay, and two types include the transitions and transversions. We'll talk about those in class. And then a nonsense mutation changes it to a stop codon. So this one then is cut off early because this mutation here caused this stop codon. Frame shift mutations are where you have an addition or deletion of a base. Okay, so if you have an extra one on here, that more than likely is going to change what the nucleotide sequence is going to code for. So here, you get a frame shift mutation where the stop codon is earlier than it's supposed to be. You could also have one, a deletion or an insertion, where it's going to change the amino acid sequence afterwards. Or if a whole codon is missing, then you would have a whole amino acid missing. Okay, it's believed that some of these have caused some diseases, such as Huntington disease, where you get a repeat frame shift mutation. There are also larger scale mutations that occur, such as in the chromosome itself. You can have part of a chromosome deleted or lost. You can have it duplicated, okay, where a sequence is doubled. You can have inversions, where they are flipped. And you can have a translocation, where one part of a chromosome can then go into a different chromosome. Interesting enough, the difference between humans and chimpanzees, one of the biggest differences is only one of their chromosomes. Most of our DNA is pretty identical, but in one of the chromosomes there is an inversion. Okay, so these lead to these mutations lead to have evolutionary effects. Mutations are, as I was saying, the starting point for evolution. Too much change, however, is usually harmful and results in selection. But not enough will also result in selection in a changing and competitive environment. So there must be some sort of balance between the amount of new variation through mutation and the health of the species. One other mutation or DNA alteration, which is very important, includes telomeres. Telomeres are specialized structures found at the end of chromosomes where they have continual or repeating events. They don't really code for anything, but it protects the ends of the chromosome from nucleases and maintain the integrity of the linear chromosome. So if you're making a copy at the very end, they would become shorter and shorter because that last little bit doesn't have a primer for it on the lagging strand. Okay, so so that's what this figure is showing. Once that primer is removed, it can't be replaced, and what will happen is you'll have a shortened template. Over time, this would become then DNA smaller and smaller, and then might get into areas where, or areas of the genes which are important for life. Okay, telomeres are composed of these repeating sequences, such as AT and AT over and over again, and telomerase then is an enzyme which makes the telomere section of the lagging strand using an internal RNA template in itself, and then allows replication to go to the very end. All right, lastly, we have DNA repair. This is errors due to replication. DNA polymerases, we mentioned before, they do have some proofreading ability. A mutagen is something that increases the number of mutations above background level. So just on their own, DNA enzymes are going to make mistakes, and there's going to be some mutations accumulating. But if there's something that causes more mutations to occur than normal, that's a mutagen. Radiation and chemicals can do this. So the importance of DNA repair is the ability to identify these mutations and go in and fix them. There are two general categories of DNA repair. You can have specific repair, which targets a specific type of lesion or mutation, and then repairs only that damage, or non-specific, which it doesn't matter what the mutation is, it just pulls it out and can fix it. So photo repair, so this mutations occur in DNA when they are exposed to UV radiation. And one of them includes a thymine dimer, where the two thymines bind to each other rather than the adenines on the other side. So a specific repair can be done through the enzyme photolyase. So it will go in and actually just fix that binding so the thymine and the adenines are bound to each other again. And it gets this energy through absorbing light in the visible range. Okay, another way you could fix this, however, is through excision repair. And in excision repair, you take whatever the damage is, and it could be a thymine dimer, completely remove the DNA, and then resynthesize it using the information from the template strand. Okay, so here, nuclease rips out the mutation. Here's the thymine dimer, rips it out, and then you get DNA polymerase to go in and repair it, and ligase seals the nick. Okay, that's all. Lots of stuff for DNA, RNA, making proteins, and mutations.